My name is Mollie Walton, and I was fortunate to have been awarded grant funding through Imprint's Opportunity Pool this year. I will be presenting my current research project entitled Effects of Genetic Polymorphisms on the OCT1 and OCT2 mediated uptake of atenolol. I completed my pediatric cardiology fellowship at Children's Mercy Hospital last summer. I'm now a second-year clinical pharmacology fellow, and through this, my research has focused primarily on the beta blocker drug class. My mentors for this project are Dr. John Wagner, who is both a pediatric cardiologist and co-program director for the Clinical Pharmacology Fellowship here, and Dr. Bruno Hagenbuch, who is the department chair of Pharmacology, Toxicology, and Therapeutics at KU Medical Center here in Kansas City. Aortic root enlargement, a manifestation of connective tissue disorders such as Marfan syndrome, is associated with aortic root dissection, which can lead to life-threatening issues. Atenolol, a selective beta-1 receptor antagonist, is commonly utilized to mitigate this risk. Unfortunately, pediatric patients continue to have suboptimal response to therapy, such as disease progression or adverse side effects precluding dose optimization. Additionally, while the Marfan patient population was the impetus for this project, atenolol is used broadly in both pediatric and adult cardiology and the management of hypertension, arrhythmias, and other conditions, thus has broad applicability. Losartan is an alternative to atenolol. A randomized trial published in 2014 by the Pediatric Heart Network, shown up on the top left, compared atenolol with losartan in children and young adults with Marfan syndrome over a 3-year study period. In this, there was notable variability in drug response within treatment groups. Genetic variation in drug metabolism may contribute to inter-individual variability seen in drug response. A follow-up investigation published in 2020, shown on the bottom right, showed variability in response to atenolol in pediatric patients to be associated with gene variants, also known as polymorphisms, within the beta-1 receptor gene being associated with variable drug response. However, there remains an absence of genetic analysis related to genes responsible for atenolol disposition. For example, the impact of polymorphisms and the transporters involved in atenolol disposition may lead to altered functionality and therefore differences in clinical effect. The human organic cation uptake transporters, or OCTs 1 and 2, encoded by the SLC22A1 and SLC22A2 genes, respectively, are influx transporters involved in the hepatic and renal uptake of atenolol, as well as the anti-diabetic drug metformin. Genetic variation in OCT1 and OCT2 is known to play a role in the pharmacokinetics of metformin. OCT2 is thought to be the major transporter for uptake of many cationic xenobiotics, such as atenolol, from the bloodstream into renal epithelial cells. A prior study published in 2015, shown here, characterized the interaction of atenolol with major renal drug transporters. Using a panel of human embryonic kidney cell lines stably expressing the various transporters, they showed that atenolol tubular secretion is mediated primarily via unidirectional transepithelial flux used in the human OCT2 MATE2K secretion pathway. Additionally, uptake of radioactive atenolol was measured in both control cells and transporter expressing cells. Compared with the control, atenolol's uptake was significantly increased in cells expressing human OCT1 and OCT2. OCT1 is a major hepatic transporter that mediates the uptake of many organic cations from the blood into the liver for metabolism. The OCT1 gene is highly polymorphic in humans. Such polymorphisms of OCT1 are associated with reduced or eliminated function, therefore decreasing transporter uptake of metformin. Slow and deficient OCT1 transport can be explained primarily by these alleles listed. The number of poor OCT1 transporters varies significantly among different ethnicities and world regions. But for example, about 9% of Europeans and Caucasian Americans are homozygous for these loss of function mutations. And while rare in East Asia, poor OCT1 transporters represent more than 80% of certain populations in South America. OCT2 is a bit less polymorphic than OCT1. At about 13%, the mutation of alanine to serine at the 270 position has the highest allele frequency across all populations and has shown more subtle but still moderate substrate-dependent decrease in OCT2 activity. Therefore, the objective of my study is to determine the role of genetic variants on the cellular uptake of atenolol, as this may affect drug disposition and clinical effect. Big picture, the rationale for this study is to provide a mechanistic understanding of variant OCT1 and OCT2 expression and function as a means to individualize pharmacologic therapy for not only atenolol, but other OCT1 and OCT2 dependent drugs, such as other beta blockers. This approach utilizes transiently transfected OCT1 and OCT2 cell lines to perform time-dependent atenolol uptake experiments, to then generate kinetics parameters of the wild type versus variant transporters. The cells that I use are human embryonic kidney or HEC293 cells. I perform site-directed mutagenesis to generate plasmid DNA for cellular transfection to assess the transporters. Radioactive atenolol is the primary study agent, and MPP, which is a known substrate of OCT1 and OCT2, serves as a positive control. The negative control is cells transfected with empty plasmids. Control cells are empty vector cells, and transporter expressing cells grow for 24 hours or 48 hours to reach 80 to 90% confluence. Uptake assays are performed at 37 degrees Celsius in a buffer solution containing cold substrates and trace amounts of radiolabeled or hot substrates. Uptake is performed at a pH of 7.4. After incubating the cells with a substrate for a specified time, uptake is terminated by washing cells with a cold buffer solution. Radioactivity of the cell lysates are then measured by a liquid scintillation counter. Protein content in the lysates is measured by their BCA protein assay, by the BCA protein assay kit. And the uptake in the cells is normalized to their protein concentrations. All experiments are performed 3 times independently to account for error. To evaluate atenolol as a substrate of OCT1, I compared the intracellular accumulation of MPP as a control and of atenolol between the HEC293 cells expressing the wild-type OCT1 and control cells transfected with the empty vector DNA. In my initial uptake experiments using transiently transfected OCT1 and OCT2 cells, MPP and atenolol uptake was less than expected compared to prior studies. In troubleshooting attempts, I compared uptake in cell lines stably expressing OCT1, shown on the right, versus transiently transfected, shown on the left. Please note that the scale on the y-axis is different, with a max of 2 picomoles per mil for stable cells and 0.6 for transient. Through these early studies, I confirmed that atenolol is a substrate for OCT1 as well as OCT2, which isn't shown here. I also found that uptake for both agents was greater in stable cells compared with transiently transfected cells. I'm still utilizing transient transfection as it's easier for this experiment, but it is important to note in comparing with published literature where stable cell lines have been used. In generating a KM, we first perform time dependency kinetics experiments. With this, you prepare a range of substrate concentrations, and we used 1 micromolar to 1 millimole to cover the expected KM range and a time point of 10 minutes. For OCT1 and OCT2, we see that uptake appears to be linear through about 5 minutes. Knowing the Km or Michaelis constant and Vmax or maximum velocity for a transporter protein is important in understanding its kinetics properties. The Km represents the substrate concentration at which the transport rate is half of Vmax. It indicates the affinity of the transporter for its substrate, meaning a lower Km value suggests higher affinity and vice versa. The Vmax is the maximum rate at which a transporter can operate when saturating amounts of substrate are present. This is determined by the number of transporter proteins available and their turnover number or efficiency. Modifications to the transporter, such as mutations, may alter these properties, which is why it's important to establish the properties of the wild-type transporter first, to then compare to the mutated transporters. To determine the Km, a Michaelis-Menten plot was generated and showed the Km of our wild-type OCT1 in our lab to be about 63 and a Vmax of 478. At a single time point and single concentration, uptake of both MPP and atenolol by the 4 OCT1 variants compared to the wild type is significantly decreased, with no significant uptake compared to their negative control. This aligns with expectations based on prior literature of OCT1 variant uptake using other substrates, such as metformin. Interestingly, the R61C uptake levels was less significantly decreased compared to wild-type OCT1, particularly in MPP. With this finding, I've been in the process of performing the kinetics experiments for this particular mutation to compare to the wild-type. Alongside the wild-type OCT1 experiments, I had performed similar site-directed mutagenesis to generate wild-type OCT2, as well as the initial kinetics experiments. However, uptake seemed suboptimal, so I performed some modifications to augment transporter expression, the results of which were equivocal. However, in the process of generating the OCT2 variants, I realized that our wild-type OCT2 was actually the more common alanine to serine mutation. So I recently reperformed the site-directed mutagenesis to generate a new OCT2. So looking into short-term next steps, I will complete my OCT1 R61C kinetics experiments, and then move into replicating all of these with OCT2 and the variants. For both transporters, I plan to quantify transporter expression on cellular membrane, because as I mentioned before, variation in kinetics can be attributable to variable amounts of transporter expression. I would like to acknowledge those who have supported me in this research, specifically the funding generously provided by the Imprint Opportunity Pool, the T32 Fellowship, and Children's Mercy, along with my mentors, Dr. Hagenbuch and Dr. Wagner, along with all of the members of the Hagenbuch Lab. Thank you.